everyone. I'm Pam Allenstein, Plant Collections Network Manager at the Association, and I will be your staff representative during this virtual conference session. Before we begin, here are a couple of tips to get the most out of your virtual conference experience. First, if you are experiencing poor video or audio quality, we suggest closing other programs on your computer, especially other browser tabs. Your inbox will still be there at the end of the session, I promise. If you have any other problems or questions, association staff are available to help. Give us a call at 610-708-3010 or send us an email at info@publicgardens.org. Second, while this session has been pre-recorded, the presenters are here with us today to answer questions, and we'll be happy to do so in a live Q&A portion after the pre-recorded session. At some point during the session, the presenters may activate a poll. Poll responses are anonymous, so please be sure to let us know how you really feel. Finally, you can submit your questions using Zoom's Q&A function that you will find at the bottom of your Zoom taskbar. Questions can be submitted anonymously, and then you can also upvote other questions to indicate that you would like to hear them answered. We will try to answer as many questions as we can during the live Q&A portion of this session. Some questions may be answered directly through the Q&A feature. Thank you and enjoy today's session.
So hi everybody, my name is Eileen Boyle. I'm the Director of Research and Conservation at Mount Cuba Center. And I'm here to talk to you today about More Than a Pretty Plant, the value of native plant trial garden. And I hope you find it really interesting. So Mount Cuba, where is it? It's in Hockessin, Delaware. And we have crafted our garden to show beauty and the value of native plants. And we protect their habitats, we care about their ecology, and we kind of want to understand it all. Now, if you look at this picture, and you can see way back over in the corner there, that's where my little trial garden is. So why do botanical gardens matter right now? I don't know, we're all kind of struggling with how they do. But I think it's really important because we can influence people to make sustainable changes and choices and measurably improve our planet. So let's do it. Why not? That's what APGA is all about. So why use native plants? Well, I like a spicebush swallowtail. I hope you do too. But that little butterfly needs to start as a little caterpillar. And if you want that caterpillar, you have to have the host plant, which is Lindera benzoin or the spicebush. So all of these kind of pyramids of life are all connected. And down at the bottom where you see the producers, that's where the native plants are. And the caterpillars are very specific that they will only eat their native plant that they've co-evolved with. And so if you take away the native plants, you lose the caterpillars, you're playing Jenga, the whole thing falls apart. So native plants and that pyramid of life is really important. So traditional trial gardens look at the attractiveness and garden performance, and they look at it in a specific area to say like, okay, is this plant good for this region? And that is wonderful, it's important, it's how we started. But as time went by, we kind of said, you know what, there's more things that we want to include in what's important. And so we have been thinking a lot about ecosystem services. And that's basically like, what does a plant give your ecosystem? And so we're thinking about pollen and nectar, nitrogen fixation, are the fruits and the seeds good for the birds? So these were all things that we kind of said, besides being pink and pretty and 2 feet tall, we want to think about these other ecosystem services as well. So I'm here to tell you that your yard matters. what you plant makes a difference. And back in 2017, Mount Cuba looked at 14 wholesale nurseries, and we said, how many of these plants that are being produced are native? And sadly, only 25%. So think of that little caterpillar, it's not getting fed, and you might as well put statues in your backyard instead of a plant. So we really want to think about this supply and demand thing. And if you look on the side here, you can see we want to create demand so that people will say, yeah, we want to buy native plants. And then the nursery guys will grow them. So we're trying to work the deal on both ends. And a trial garden can really help that. So, ooh, isn't that pretty? Well, we're more than a pretty plant. This is the trial garden at Mount Cuba Center. And this is our Baptisia trial. So what do? Well, we basically take native plants and their related cultivars, you can call them nativars if you want to, hybrids, and as many straight species as we can have, and we go out there and we trial them, and we see how they grow. So let me talk about the trial garden a little bit. It's been here for, I don't know, 12, 13 years in its newest form, and there's a sunny spot that's about 10,000 square feet. There's a shade structure with kind of nice overhead shade cloth, and that's about 5,000 square feet, and that's our Hugo trial, which was quite pretty. And our staff, right now we have Sam Hoadley, who is our manager of horticultural research, and formerly we had George Coombs, who is now happily our director of horticulture. So they've both been really important in developing the trial garden with me. So why do we do this? Well, I told you about the why of native plants. We want to inform consumers that they do have choices. We want to introduce those underused species to the nurseries, as well as tell other botanical gardens and the world about, you know, why should you plant these things? So how do we do it? Well, we look at a genera, so say like the genus Phlox, and we go with the straight species, the hybrids, the cultivars. We look at everything that's native to the eastern United States. That's our nativity kind of lens. We keep maintenance to a minimum so that we really don't want to, you know, fluff the plants. We just water them when they are almost dead and throw some compost, and that is it, because that's probably what a homeowner's going to do. And then we look at an ecological question associated with that plant. So it's more than a pretty plant. And we do all of this for 3 years. Audience. Who's our audience? Well, it's anybody who walks through the gates at Mount Cuba, and everybody beyond the gates too. So our visitors, the nursery industry, homeowners, researchers, retailers, anybody that will think about plants. And this is a picture of our Helenium trial that we just recently completed. So once a year we have a trial garden event, and we invite anybody who we think could be an influencer, the nursery industry, garden writers, horticultural professionals, and if anybody wants to come, you can contact me and we'll invite you too. And we always have an ecological question. So this is an example of the carex that we're working on right now. You probably all know carex, sedges have edges. Well, they're better than grasses in the shade. They have a horticultural use, but they have a conservation use as well. And so we have a memorandum of understanding with our Delaware State botanist and DENREC, and he brings us seed all the time. And we grow stuff for him for conservation, and he helps us with the horticultural stuff. So this was a wonderful kind of question. Well, if we take these carex and we put them back out in the wild, will they do any good? Well, here's endangered pug turtles nesting on the carex that we grew. So it made a big difference. And we're always kind of answering both the conservation and the horticultural questions at the same time. So how do we do this? We take data collection, and there's George with his little computer going out there, and he measures everything. Well, not him anymore, now it's Sam. But we measure the things. We say how tall, how wide. We look for pest and disease observations, because if it ain't pretty, nobody's planting it. So that's, you know, kind of the first lens. And then we assign ratings of, 1 to 5, 1 being terrible, 5 being really good. We look at garden quality, foliage, habit, is it floppy, does it stand up by itself, floral display, and all the usual suspects. But then what we do beyond that is we have a pollinator watch team, which is 24 dedicated citizen scientists that get trained, and they go out with their little clipboards, and we give them a pollinator watch data sheet. And what they do then is each of the plants has a little number and label on it. So for example, echinacea is number 1 is Cheyenne Spirit. So they go out there with their clipboard and their piece of paper, and they go to one plant and they observe that one plant for 60 seconds. They have a little timing device, and they look at that targeted pollinator. Now for different trials, it might be a butterfly or a hummingbird or a bee or something different. So they look at that targeted pollinator, and then they collect the data. At the end of the year, we take all the data, put it together, and we do this over multiple years to make sure we're getting a true data set. So that's pretty cool. Then we have, it's pretty, it's pink, and here's its pollinator. So what have we done in the past couple of years? I've been at Cuba for 15 years, starting with the trial garden, and we've completed, quite a few trials of Selenium, Phlox, Monarda, Baptisia, Coreopsis, Echinacea asters. We're working on Hydrangea, Amsonia, Echinacea 2, which is because there's so many darn cultivars that are coming out, you can't keep up with them. And then Vernonia, Solidago ferns, and Clematis are coming up. So what we then produce, and I'm going to hold up my little, is this beautiful, nice final research report in a published form. Then we also have it on the website, and you can see the website summaries. And at the end of the talk, I'm going to show you where to go to access all of these things. So like I said, if we wanted to do phlox, we would like to do all this research, and we'd say, what are all the phlox cultivars we can find? And what are their hybrids? And what are the native phlox that aren't, on anybody's radar? And we'd put them all together. And that's what we do for each trial. So from 2015 to 17, we trialed phlox to test their powdery mildew resistance. There's a whole bunch of new cultivars. We wanted to look at the nectar for pollinators, and there were a bunch of new species that were out there. So we wanted to, not new species, new cultivars. We wanted to say, how are they holding up? Because sometimes people, when we did Heuchera, there were like all these different cultivars, and they all looked exactly the same and didn't perform as well as maybe they should have. So, we're kind of a little bit of a reality check for the industry to kind of say, like, yeah, here's what grows well here. So there were 94 different accessions in 8 different species, and we did a sun-loving one, and we did a shade phlox trial. So the kinds of things that you find is, here's Phlox amphifolia. Now, I didn't even know what that plant was until I had that trial, and it has a lot of powdery mildew resistance and a lovely little plant that's just not in the trade. So how do we do this with just 2 people? Well, we have fellows, Mount Cuba fellows. We have interns and students. We work with University of Delaware a lot. We've worked with Doug Talamay and Dr. Deb Delaney. And we use our own interns as well. And this is a picture of Owen Cass, one of our Mount Cuba fellows. And we were thinking about, well, we want to talk about nectar on both Monarda and the phlox. So how the heck are we going to do it? So we made up this kind of crazy way of taking a little pipette, sticking it in the flower, pulling out the nectar, and then using a refractometer, which just tells you how much sugar is in something. And so that's what you're seeing here. And we were thinking, oh, maybe because the cultivars are different, certain butterflies like this, and maybe it's the sugar. Turns out it wasn't. Pretty much all the cultivars had the same amount of sugar. So we kind of went down that rabbit hole, but we did find that the butterflies had definite preferences. So we looked and we watched and we said which of the different butterflies are landing on these different flocks. The pollinator watch group did. And they came out with this astounding results that Phlox paniculata genera had 5 times more pollinators than any other plant. So it had no powdery mildew. It won like the prize. So if you want to plant a phlox, I suggest genera. We don't know why they like it so much, because it's not the sugar. It might be that it's a smaller flower that is kind of more like a lilac kind of shape, so they could get more bang for their buck with their little proboscis. But we don't really know. I've had people tell me that, nicotine came from plants, so there might be like some butterfly crack in here that we don't know. But this plant attracts butterflies like something you have never seen. So that was a success story. And sometimes we, go and try things, and like the sugar content, it really wasn't the answer. But that's what science is about, and that's why we do research. So we also did Monarda for powdery mildew, new dwarf cultivars. We did 40 accessions and 6 different species. And we did this one because you probably heard of Monarda didyma and fistulosa, but there are a whole bunch of other ones out there that nobody's heard of. So the Bradburyana, the punctata, the clinipodia, and the citrodora were ones that were like, oh, these look cool, so let's try them. And what we found was this Monarda bradburyana, the eastern bee balm, was really a beautiful plant. It's a little floppy and a little sparse, but we've collected seeds and we're trying to develop a better strain of it, and to release to the nurseries. The Monarda punctata was also a wonderful pollinator. It was more of a biennial to not truly perennial, but we found out a lot about it. Again, the pollinator watch team went out and they counted hummingbirds, and they counted moths and butterflies, and the winners were Jacob Klein and Claire Grace, which were also, again, like the phlox genera, they were our best growing plants as well. And there may be a correlation that if something is healthy and doing good, it can really provide for your pollinators as well. Baptisia, I love Baptisia. Everybody should plant it. It's not very popular because it's hard to germinate, slow to grow, and when you buy it in a store, it's like this big, and you're like, that's not too impressive. But you wait 2 or 3 years, and it's like a peony. Like, it gets better every year. So it should be more popular. There are new cultivar releases out there, and we wanted to also test the habit, because some of them can be 5 feet tall, some of them are little pygmies. So we did 46 different accessions, and look at that, isn't it pretty? I loved that trial, and the bees go crazy for it. It's a really cool plant. So the wildlife value is high. It's a host plant for elfins and tailed blues and hoary edged and spotted skippers, sulfurs, all kinds of butterflies and moths. And it's great for bumblebees too, and you can see that picture of the bumblebee kind of opening up that pea-like flower and going in and getting plenty of pollen. We looked for their ornamental piece as well, for the gray stems, the bluish foliage, the excellent fruit. And, if you want to read our little thing here, you'll find out all about each plant and everything about it. Baptisia lemon meringue, look how pretty that is. Was a really nice one, a vigorous grower, base-shaped, kept going for 3 weeks. Here's a smaller one called Blueberry Sundae. So, we go through all the cultivars, we grow them side by side. We see, does it stand up? Is it a flopper? What are the bees, the birds, the butterflies doing? And this one was really interesting because when we dug it up, you see those little round dots on the picture. It's the nitrogen fixer. So it's taking atmospheric nitrogen and with bacteria, rhizobium, turning it into nitrogen that the plant can use. So it not only is it beautiful, it makes your garden healthier, and you don't need fertilizer whatsoever. Coreopsis was an interesting one. There's a lot of breeding going on in Coreopsis these days, and some of them are annuals, a lot of them are annuals, and we wanted to see, were they hardy? And we also had some wild collected materials of species that just weren't in the trade. So this was a big trial from 2012 to 14. It was actually 3 years. We had 94 accessions, 67 perennials, and 27 annuals. And here is a picture of the trial garden with all of the annual Coreopsis. So all those different colors, we were like, well, Coreopsis is usually yellow, and we're not vegan about native plants. We're like, we want it to be prettier. We want more people to use them, and so we have to see, are they as good from an ecological standpoint? So we looked at this, and I was out there one day looking at all these annuals, and they're red and they're pink and they're different colors, and there were different bees on each different color. And I was like, what's going on here? So that, made us decide to look at the flowers and the flower types. So here's Owen, and he's got his little dust buster, sucker upper machine that basically sucks up the bees. And then you can look at their diversity across different cultivars. So the vacuum sampling was quite interesting. And what it showed us is that by changing all these different colors and increasing the variety of the colors, you got more different kinds of bees than just one little yellow plant. So there are sweat bees and wasps and bumblebees. But we also kind of wanted to look at it and say, well, is our hypothesis true? Do you think that the colors might have changed it? So here's a whole bunch of different little Coreopsis. And if you look, just for the sake of argument, at the top 5, so these top 5 are all yellow. And if my hypothesis was true that, like, okay, yellow flowers would have the same bees, we could test that. Wasn't true. Color didn't seem to actually work, and I'll show you in a minute the results of that. But if you look at these flowers, they're ray and disc flowers. So the rays are the outside, like kind of the sunshine, and the inside is the little disc. So what we think is happening is the size of the disc was saying, oh, I'm a sweat bee and I can fit on this. So it was really kind of a very interesting thing that we found out. So think of those 5 yellow flowers. Now we're going to the next slide, and we're going to look at bee diversity. So here, these top 5, they're all different. So if it was the color, they would be the same. And so we found that the composition of the insects changed as you changed the color and the size and the whole thing, but it increased the diversity. So people that are like, oh, cultivars are no good and we shouldn't mess with nature, not necessarily true, because this is showing us more diversity. So as I said, here's Coreopsis stripteris, gold standard with George. And the point of this is we do plant introductions that come out of these trials. And we have introduced 17 cultivars to the trade since 1988. And some are in the pipeline, and many more are being evaluated. So just as we have coming trials, we'll have coming plant introductions for you guys to see. One of our newest plant introductions that was just released in 2020 is Iris First Color Purple Flame. And we've done this with North Creek. It has these beautiful purple leaves in the spring. And besides the Coreopsis tripteris, we've done Solidago's Golden Fleece, which is a wonderful little goldenrod, and plant introductions that have standed the test of time. So Purple Dome, Bluebird, Chocolate. And when you have a trial garden, what's great about it is you're able to really look at something for a while to say, yeah, this is a really good plant, and we want to reintroduce it as a cultivar. So we have other plant trials coming, like the hydrangeas and the echinacea, and I'll have to come back another time and talk to you about those. But you know, they're going to be exciting. We're going to look at double flowering in echinacea and pollination on the hydrangeas. So it's going to be really cool. But you know, this is only the beginning. All these people that are studying plants should find the best native plants and encourage their use. And so we encourage you to come to Mount Cuba when this is all done. But if you can't, go online to mountcubacenter.org and let's click on the research button, and then you can go to Trial Garden, and all of our various publications are available both in published form and as well as web descriptions. So thank you for listening to me about our trial garden. We love it, and we hope to see you at Mount Cuba Center soon. Okay. So I think I answered everybody's questions, but if anybody else wants to ask me anything about, you know, what we're doing, there are a couple exciting things that are coming up soon. We have our hydrangea trial is looking at lace cap versus the pom-pom ones, and the lace caps are winning pollinator big time. We're also looking at more carex, and the double flowered echinacea should be coming. We've got solidago, and also we're looking at the solidago, all the different species. There's a whole lot of them. So we're looking at the hydrangea radiata, which is the lace cap one, so that the outside part is the sterile flowers and the inside is the fertile ones. And we have found that something like Annabelle, which, you know, is kind of what's out there, it doesn't really have that many pollinators at all compared to what we have in the way from the hydrangea radiata. And we worked with Michael Durr and got his collection of hydrangeas, and so we've been growing those all out too. So that's going to be an interesting trial. We haven't, there was a question, any container garden trials looking at pollinator diversity? We haven't tried that. It is a good idea. But you know, we're limited by, we have one full-time person, one part-time person, and then our army of volunteers. So the question was, do your volunteers look for caterpillars as well as butterflies? We really haven't, but if it would be a host plant, because it depends on if it's a host plant or a nectaring plant. So those are kind of two different animals there. But it's a good idea, like say we did, you know, any of the Asclepias, it would be really good to look on that. We didn't analyze, the question was, did you analyze the chemistry of the nectar sources? We did not, and anybody who wants to help me with Floxigena and look at what's in that would be great, because we need to figure that one out. Are you looking at pollinators diversities? Us, the life of the bloom cycle. Yes, indeed we are. From when it first starts to when it's done, and they come out, and measure it for a certain amount of time. They fill out their forms. And so we also can look at what the weather was, and then we average it across 3 years. Have you considered being part of Nativars research project through Budburst? Again, really good idea. I would love to, but maybe if I get another person. Do I know of other gardens in the US or Canada doing similar trials? Well, we have worked a little bit with Chicago, talking to them. They kind of like started the whole trial garden thing, and they are doing, have bred some baptisias and done some really good work with natives. So we have worked a little bit with them. Other than that, I'm not, other than at some universities, I know that Penn State did a cultivar trial on pollinators as well. Do you include a control group of non-cultivars alongside the cultivars? Yes, we do. If we're doing we'll do straight species, so that hopefully we have the parents of the cultivars. Sometimes you don't know from whence the cultivars came. Our definition is we use half of, it has to be half native. So, if it's a hybrid, we wouldn't put a non-native plant in there, but we do put the straight species. Has the nursery industry in your region used the results in choosing what cultivars to sell? That's why we did that, where you said it matters in your own backyard. We did Mid-Atlantic nurseries, and we said, okay, how much native did you use and buy? And in 2017, we'll continue to do that to see if the needle shifts at all. Also, when we have our nursery shindig, where it's a trial garden and everybody comes to the party, they all come and take notes, and they see what's in bloom, and it really affects them, what they choose. Do implemented nativars need to be highly localized from the perspective of conserving the local gene pool? Well, we would love everybody to use seed and, maybe, not have to push cultivars, but the problem is people only will buy what's in the store, and cultivars will only be in the store because nursery guys are not going to produce seed plants. So we try to do cultivars that are not highly bred, so that they're just natural selections, so that hopefully the gene pool can not be so artificial. Just a note to thank Cuba for the wonderful plant trials. Their team is excellent. Oh, thank you. This is from Brian Trader. He used a publication in the selection of plantings for the Delaware Botanical Garden. That's really cool. All right, are you growing all the plants on site? If not, are you screening to avoid plants that might have been exposed to neonicotinoids? We buy plants sometimes from nurseries that we know. We do grow a lot ourselves from seed, and they, like we've been growing the Solidagos that are going out in the trial for a year. So hopefully by the time they actually go in the garden, there are no neonicotinoids in the plant. So we're concerned about that as well. The Dallas Arboretum has significant trials, including 3-year perennials. Wonder if they'd be interested in partnering. You have my contact information, and I am really open to helping anybody that I can, and get going, whatever we can after this pandemic is over. So that looks like, are there any more questions? Doing okay? Got all the questions answered? that all right, Pam?
Excellent. That was wonderful. And thanks for all those fantastic questions too.
Yeah, they were great.
Serious interest in this topic, and you did a great job sharing the wonderful work that Mount Cuba is doing.
And everybody come visit us.
Absolutely, absolutely. We're lucky. We live right down the road from you.
That's right.
So we can visit once we make a reservation to get into the right time slot, which is all good.
Right.
So if folks have more questions for Eileen, she's provided her email address, and you also have the contact information from the presentation. Please join me in thanking Eileen for this excellent presentation, this excellent session. And we're going to wrap up this session now. If you'd like to keep the discussion going, please click the live discussions tab on the conference website and start a topic thread. And now in just a couple of minutes at the top of the hour, please join us for our special session on how to waive admission, deliver mission, and keep the lights on before we wrap up today with the virtual conference send-off. Thanks again for attending, and see you at the next session. Bye.